Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series. And the question for today is discuss the technique of producing a virus free nucleus stock in potato. So let's see how we can approach the answer, and this is a PYQ asked in the IFOS exam last year. So let's see how we can approach the answer. So as from the question you can understand that we have to produce virus free nucleus stock of potato. What are the techniques through which we can produce virus free nucleus stock of potato? So this clearly suggests that potato is highly infested from virus. Okay, that is why we are going to produce the virus free stock. So in the introduction part, you can simply mention about those viruses, how those viruses are affecting potato. So simply you can mention in the introduction part that potato is a vegetatively propagated crop, vegetatively propagated crop, and it is highly susceptible to degeneration due to accumulation of virus, and it is highly susceptible to degeneration due to accumulation of viruses. And you can give example of such some viruses. I am giving you the names. PYV is one virus, PVX is one virus, okay, PLRV is one virus. So these are virus, and I think if you have read about a disease, that is potato mosaic virus disease, potato disease, mosaic virus disease. So this is from the virus only. It is one of the most affecting diseases of potato due to virus. So you can give examples of these virus: PPY, PPX, PLRB. You can write full form also, but that is a kind of complex names. So you can simply use these simple forms also. So it is good enough. Okay. So therefore production of virus free nucleus stock in potato is essential for getting good quality seed production and to maintain varietal purity. So that is how you can introduce the question. Now we will move to the techniques, how we can produce virus free nucleus stock in potato. So first of all, we will try to select good mother plant. Selection of mother plants. So mother plants should be true to type. They should be healthy, and they should be collected from disease free areas and high altitude locations. Disease free areas. And generally, if you see the diseases of potatoes in high altitude, the disease affected to potatoes are less as compared to lower altitudes. So simply you can write disease free areas. In example, if you want to give disease free high altitude areas. So there should be no symptoms of any disease, no viral symptoms, and the desired variety should be well upheld. So you can select a good mother plant there. Then you should also do the virus testing of mother plants. That is also important technique which should be done. Virus testing of mother plants. So there are various methods through which we can do virus testing. For example, you can write about here ELISA. You can write about PCR testing. We can write about indicator plant test. So these are the some of the tests which can be taken to produce virus-free plants. So not produced to test that the mother plant is virus-free. Next, we can do meristem culture, one of the most important steps. Not steps, or you can say techniques. One of the most important techniques. You must write this technique whenever writing this answer, because meristem culture is one of the best methods to produce virus-free plants. So here generally you see that we can do meristem culture because meristem tissues are generally free of virus due to lack of vascular connections. As they are, meristem tissues are free of virus due to lack of vascular connection. So this can be used meristem culture. Then you can do macro propagation, another very good method. These two must be written in your answer. There are no substitutes. Okay. So micro propagation, here the virus free plantlets are multiplied using in vitro propagation, that is in our monitoring in the lab, in vitro propagation. So here we can use nodal cuttings in tissue culture, use nodal cutting in tissue culture. We will try to maintain aseptic conditions, as it is easier to maintain as we are growing in laboratories. So these two must be mentioned. Then we can do virus indexing of regenerated plantlets. Virus virus indexing of regenerated plantlets. So each regenerated plantlet is tested again for virus presence using ELISA or PCR. So after the propagation, we will again test using ELISA, PCR. So all the virus plantlets should be tested. Okay, on the all the plantlets should be tested to be virus free, and only index negative plantlets are selected. Only those plants which show negative on this test are then selected. Next technique we can use is hardening and assimilation. Hardening and acclimatization. Acclimatization. So here tissue culture and plantlets are gradually hardened. How they are hardened? So hardening is a process where we use controlled humidity, controlled light. Okay, in a poly house it can be hardened. Controlled humidity, controlled temperatures. See what happens. Suppose this is lab, this is the poly house, and this is the field. So what happens? In the, if you have been to hospitals, if you see some serious patients, they are, when they are discharged from operation theater, okay, from the operation theater, they are just first kept in ICU unit. Okay, ICU unit, because they need intensive care. So here the labs are the ICU unit, and after that ICU unit they are transferred into HDU unit, where there is certain kind of hardening. So in this HDU unit there is certain kind of hardening. Okay, in HDU unit there is a although there is everything is available there, but mainly patients are kept without oxygen, without that much of kind of support, so that they can try to overcome their disease. And after HD unit they are sent to general unit. Okay, general unit. So in these two units the process which happens is kind of hardening. The patient prepares himself for the outer environment which he have to face. Outer environment. So simply happens for the potato here also. The potato in the lab after being produced is shifted to greenhouses, polyhouses. These two let it be polyhouses. Polyhouses or controlled field conditions, so that they get hardened, they get able to they get able to be able to survive in the outer fields. Okay, so this is what you should know. So this is what the process of hardening and accumulation is. So first of all they are soil, they are kept in soil-less medium, okay, which control humidity and light, and later shifted to greenhouse or net house for assimilatization, and after that they are shifted to the normal fields. So this is, you should know, hardening and assimilation. I hope this example helps you. Then you can use the production of nucleus stock. So all the virus-free plants which are hardened and assimilized, okay, are when grown in insect fruit net houses, then these plants are monitored for off-type and virus symptoms. Okay, so that there is no off-type presents, there is no virus symptoms present. You should be testing for all these, monitoring for all these. Okay, so this is the need. Then we should keep doing regular rugging and monitoring, rugging and monitoring. So rugging, you simply know, is the removal of off types. So we will keep continuously removing the off types, and periodic visual inspections, testings, all this should be done to maintain the or ensure the stock purity. So these are the things which you can mention in your answer, and I think it would be a fair enough answer. And simply in conclusion, you can mention about the meristem culture and micro propagation, how they ensure healthy seed material, maintains genetic purity, and enhances seed multiplication. And this forms the base for foundation and certified seed production. Okay, so these things can be mentioned in your conclusion part. So this is all for today. I hope you understand all the points, and I think you are able to understand the things. So that is all for today. If you like it, please press the like button, please do share, and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis, and if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.